Welcome to the deep dive. Today we're looking at something absolutely fundamental, something we usually take for granted, the cell membrane.
Right. It's easy to picture it as just, a bag holding the cell together.
Exactly. But it's so much more than a static wall. Our sources paint this picture of a really dynamic gatekeeper. Think about it. Every cell needs nutrients in and waste out.
Absolutely critical. Without that constant traffic control, well, life just wouldn't work.
So that's our mission today. We want to understand how cells manage this flow. What are the rules? What are the mechanisms? We've got some great material outlining these processes, and we're ready to unpack it all.
Sounds good. Let's dive in.
Okay, so when we talk about what gets in and out, there's a core concept, right? It governs this whole traffic control idea.
Yeah, and that concept is permeability. It's really the starting point. The research we looked at breaks it down nicely.
How so?
Well, you can think of three basic types. You could have impermeable, like a solid wall, nothing passes.
Okay, like a sealed container.
Pretty much. Then there's freely permeable, everything gets through, like a net with huge holes.
No, selective.
Not at all. But the key one for cells, the one that makes everything work, is selectively permeable.
Selectively permeable, meaning it chooses.
Exactly. It chooses. It's like a very discerning border guard. This is crucial for the cell to maintain its own special internal environment, distinct from the outside world.
And it's not just picking things at random, I assume. There have to be specific criteria for this choosing. What determines if a molecule gets the green light?
That's right. It's not random at all. The sources point to four main things the membrane looks at. First, just the size of the molecule. Is it small enough to fit?
Makes sense. Too big, no entry.
Then there's electrical charge. Ions with positive or negative charges behave very differently trying to cross that membrane compared to uncharged things.
Okay, size and charge. What else?
Molecular shape is another one. Sometimes a molecule might be small enough, but if its shape is wrong, it won't fit through a specific channel or interact with a carrier protein correctly.
Like a key needing to fit a specific lock.
Precisely. And finally, a really big one is lipid solubility. Can the molecule dissolve in fat? Because the membrane itself is largely made of lipids, right?
Right, the lipid bilayer. So if it's liquid soluble, it can kind of sneak through.
It often can, yes. It makes passage much easier. So size, charge, shape, and lipid solubility, those are the key gatekeepers.
Got it. So we have this super selective dynamic membrane making decisions based on these factors. Now, how does the actual movement happen? The sources seem to split transport into two main strategies.
Yeah, two broad categories. It really boils down to energy use.
Right. You've got passive transport, which apparently needs no energy from this cell, kind of like floating downstream.
Exactly. It just happens if the conditions are right.
And then there's active transport, which is the opposite. It does need energy. Like paddling upstream, you need to put in effort, usually ATP, right?
That's the one. Active transport requires ATP to power the movement, often against the natural flow. This passive versus active distinction is fundamental.
Okay, let's tackle passive transport first. No energy needed. The most basic form seems to be just diffusion.
Diffusion. Yeah, it's the simplest. We see it all the time. The sources describe it as just molecules mixing randomly, a solute spreading out in a solvent.
Like dropping food coloring in water, or the classic example, perfume spreading in a room.
That's the perfect analogy. You spray it in one spot, high concentration there, low everywhere else, and naturally, randomly, the molecules spread out until they're evenly distributed. The concentration gradient disappears.
And the absolute key rule here, it seems, is that things always move down their concentration gradient in diffusion, from high concentration to low.
Always. It's a fundamental physical principle, like a ball rolling downhill, as you said. It doesn't need a push, it just goes.
It's amazing how cells harness that, just letting physics do the work.
It really is. Simple equilibrium driving vital processes. Cells are basically leveraging the universe's tendency towards randomness to get stuff done efficiently. Saves a lot of energy for harder tasks.
Okay, so within passive transport, there are different types. The first is simple diffusion, the easy pass, you called it. What kind of stuff gets this easy ride right through the membrane?
According to the material, it's mainly two kinds of things. First, lipid-soluble compounds, things like alcohols, fatty acids, steroids, because the membrane is lipid.
They just dissolve right through.
Pretty much, yeah. They can navigate that fatty core. The second group is dissolved gases. Think oxygen and carbon dioxide.
Crucial for breathing, for respiration.
Absolutely vital. O2 goes in, CO2 comes out, just by simple diffusion across the membrane. Effortless gas exchange.
Okay, but lots of important things aren't lipid-soluble or tiny gases. What about them? The sources mention channel-mediated diffusion. Sounds like special doorways.
That's a great way to think about it. For things like water-soluble compounds and ions, which can't get through the lipid part.
Because oil and water don't mix, basically.
Exactly. So the cell uses transmembrane proteins, proteins that go all the way through the membrane to form specific channels or pores.
Like tunnels.
Tiny tunnels, yeah. And these channels are often incredibly specific. A channel might only let potassium ions through, or only sodium ions, or maybe just water. It really reinforces that selective permeability idea again.
So even the doors are selective, not just any key works.
Definitely not. The size and charge inside the channel usually restrict what can pass.
Okay, channels help things that can't dissolve through. Then there's another layer, right? Carrier-mediated transport, but still passive. So, facilitated diffusion. This sounds more complex, like a bouncer checking a list.
Yeah, the bouncer analogy works well here. This is still passive, so it's still moving things down their concentration gradient, no ATP needed, but it requires a helper protein, a carrier.
And why is this needed? What kind of molecules use this?
This is essential for molecules that are, frankly, too big to fit through those channels. Think crucial molecules like glucose, the cell's main fuel, or amino acids, the building blocks for proteins.
So how does the bouncer work? The carrier protein.
Okay, so the molecule, let's say glucose, approaches the carrier protein. There's a specific receptor site on the protein that glucose fits into perfectly, like a docking station.
Specificity again.
Always specificity. When glucose binds, it causes the carrier protein to change its shape. It kind of flips or rotates, opening a path to the other side of the membrane.
And then it releases the glucose inside.
Exactly. Once the glucose is released, the protein snaps back to its original shape, ready for the next passenger. It facilitates the diffusion, hence the name.
Got it. So simple diffusion, channels, and facilitated diffusion, all passive, all moving downhill concentration-wise.
Correct. All driven by the gradient, no direct energy cost to the cell.
But what happens when the cell needs to move something against that gradient? Like pumping something out even when there's already a lot outside?
Yeah.
That's where active transport comes in, yes.
Precisely. This is the game changer. Active transport moves substances against their concentration gradient, from low concentration to high.
Swimming upstream.
That's the perfect image. And just like swimming upstream, it requires energy. The cell has to expend ATP to make this happen.
And it still uses carrier proteins like facilitated diffusion.
Yes, it does. Active transport is also carrier-mediated. It needs those specialized protein movers or pumps embedded in the membrane.
The sources mention ion pumps and exchange pumps. The big one seems to be the sodium-potassium exchange pump. Sounds really important.
Oh, it's incredibly important. Probably one of the most critical pumps in our bodies. It's constantly working in almost all our cells.
So what does it do exactly? How does it work?
Okay, its job is to maintain specific ion gradients. For every cycle, it actively pumps 3 sodium ions, Na+, out of the cell.
Okay, 3 sodium out.
And in exchange, it pumps 2 potassium ions, K+, into the cell.
3 out, 2 ends, against their respective gradients.
Yes, usually both are moving against their concentration gradients. Sodium is typically higher outside the cell, potassium higher inside, so it's pushing them where they don't want to go passively.
And the cost. You mentioned ATP.
Yes. Each full cycle 3 sodium out, 2 potassium in, costs the cell 1 molecule of ATP. That's the energy source that powers the pump's shape changes to move the ions.
1 ATP per cycle.
Yeah.
And this is happening all the time.
Constantly. In fact, this single pump can consume a huge chunk of a cell's total energy budget, maybe 20–40% in some cells, like nerve cells. It's a massive energy investment.
Wow. So what's the payoff? Why spend all that energy? What does this constant pumping do for us?
The payoff is enormous. This pump is fundamental for, well, almost everything. It maintains the electrochemical gradients across cell membranes, which is absolutely essential for nerve impulses. Your ability to think, feel, see, it relies on this pump.
So brain function depends on it.
Critically. And muscle contraction, too. Your muscles wouldn't be able to contract without the proper ion gradients established by this pump. No movement, no heartbeat.
Heartbeat, too. Good grief.
And it also helps regulate cell volume, preventing cells from swelling up or shrinking too much. So nerve function, muscle function, basic cell integrity. It's indispensable. It's staggering. This invisible, constant work. Just mind-blowing.
Right.
Okay, so let's pull back a bit. We talked about facilitated diffusion, passive, and active transport both use these carrier proteins. The sources say they share some common features.
They do. There are three key characteristics of carrier-mediated transport, whether it's passive facilitated diffusion or active transport. First is specificity. We touched on this.
One carrier, one job, basically.
Pretty much.
Yeah.
One transport protein is designed for one specific substrate or a small group of related substrates, like the glucose transporter only moving glucose.
Okay, specificity. What else?
Second, they show saturation limits. This means there's a maximum rate of transport because there's only a finite number of carrier proteins in the membrane, right?
Like checkout lanes at a store.
Exactly. Once all the carriers are busy moving molecules, the transport rate can increase. Even if you add more molecules waiting to be transported, they're saturated.
That makes sense. Unlike simple diffusion, which just depends on the gradient. Correct. And the third characteristic is regulation. The activity of these carrier proteins can often be controlled. Hormones, for example, can influence transport rates.
So the cell can turn the transport up or down based on its needs.
Precisely. It adds another layer of control, allowing the cell to adapt its transport activity to its current metabolic state or external signals. Specificity, saturation, regulation, key features.
Very cool. Now, one more thing about these carriers. The sources mention co-transport and counter-transport. Sounds like they can move more than one thing at a time.
Yes, some carrier proteins are designed to move multiple substances simultaneously. Co-transport, which is also called symport, is when the carrier moves two substances across the membrane in the same direction at the same time.
Like two people going through a revolving door together.
Kind of, yeah. Often, one substance moving down its gradient might pull the other one along with it, even if that second one is going slightly uphill.
Okay, that's co-transport and counter-transport.
Countertransport, or antiport, is when the carrier moves two substances in opposite directions. One goes in while the other goes out.
Ah, like an exchange.
Exactly. The sodium-potassium pump we spent time on, that's a perfect example of active countertransport. 3 sodium out, 2 potassium in, an exchange powered by ATP.
Wow, okay. We have covered a lot of ground.
Oh,
From simple diffusion, just letting things spread out,
like the perfume.
all the way to these incredibly complex, energy-hungry pumps like the sodium-potassium pump that power our nerves and muscles. It really drives home that idea we started with. The cell membrane isn't just a wall.
Not at all. It's this incredibly sophisticated, dynamic, selectively permeable gatekeeper. Constantly active, constantly regulating.
Managing traffic with just amazing precision.
And it's crucial to remember this isn't just some abstract biology concept. This dynamic transport is the basis for everything cells do, maintaining balance, getting energy, communicating, responding, surviving.
It really is the foundation.
It underpins all of life, really, that constant controlled exchange across the boundary.
So as we finish up this deep dive, here's something to think about. If this transport is so precise and so vital, what happens when it goes wrong? How might tiny disruptions in these transport systems lead to disease? Or, looking at it another way, how could we potentially target these transporters for new medical treatments or drug delivery? And maybe even more fundamentally, what does this ceaseless invisible traffic across every cell membrane tell us about what it actually means to be alive?
Profound questions. Definitely food for thought.
Absolutely. Thanks for joining us on the deep dive. We'll see you next time.